Hi everyone, thanks so much for joining us today. We're going to get started at noon Eastern time, so we'll start the seminar in just a few minutes. All right, we're going to go ahead and get started. So hello everyone, thank you all so much for joining us today. Welcome to the NOAA Omics Seminar Series. I'm your host, Catherine Egan. I sit in NOAA Ocean Exploration, and I'm joined today by my co-host, Nicole Miller. Nicole, would you like to introduce yourself?
Hi, everybody. I am the new Omics coordinator within Ocean Exploration. I will be taking over this series from Catherine Egan from this point forward. Thanks, Catherine, for everything you've done to get us started on the series, and I look forward to hosting in the future.
Thanks so much, Nicole. Really appreciate you taking over the series, and the folks who regularly attend are in good hands, and I hope you all are looking forward to our continued great lineup of speakers. All right, so with that, a quick introduction on omics. Omics, which is a suite of tools used to analyze DNA, RNA, proteins, and metabolites, has become a large priority here at NOAA in the last several years. We established this seminar series in an effort to increase transparency and collaboration, highlight the incredible omics-related research currently underway, both within and outside of NOAA and with our NOAA partners. You can view past seminars on the Omics website, which is linked in the chat right now. As participants, you are muted, but feel free to type questions into the questions for staff box throughout the presentation. At the end, I'll read the questions for our speakers to answer. So with that, our presentation today is titled Microorganisms and Metabolites as Diagnostic Indicators for Coral Reefs. Our speakers are Drs. Amy Apprill and Elizabeth Kujawinski from the Woods Hole Oceanographic Institution. All right, so Amy, I'm going to go ahead and give you screen sharing permissions now. And while you are getting set up, I will go ahead and introduce you to our audience. So Dr. Amy Apprill is a microbial ecologist who received her MS and PhD degrees from the University of Hawaii. She has over 20 years of experience studying coral reef ecosystems. She received the Early Career for Environmental Research Award from the American Society for Microbiology in 2020 and is a team leader of WHOI's Reef Solutions Initiative. I have a website for that, which I'll link in the chat in just a moment. And we're also joined today by our other speaker, Dr. Elizabeth Kujawinski, who is a marine chemist who received her PhD degree from the MIT WHOI Joint Program in Oceanography. Her lab has led the development of metabolomics approaches for marine microbial investigations. She's a director of the Center for Chemical Currencies of a Microbial Planet. I also have a link for this too, which I'll put in the chat. So thank you so much, Amy and Liz, for presenting today. Amy, I will now pass it off to you. And before we do that, I can see your screen and it looks great.
Wonderful. Thank you so much, Catherine, and thank you for Nicole for taking over the organization of this great seminar series. So indeed, I'm Amy Apprill. I'm going to be taking you through the slides that I prepared today along with Liz Kujawinski, and Liz is primarily going to be here to correct anything inaccurate that I say, as well as answer questions at the end too. So you have a certified microbiologist and a certified chemist on this call for your questions today. So I'm really excited to tell you about some of the work that we've been doing using microorganisms and metabolites, working on developing them as diagnostic indicators for coral reefs. Before I get started, I just wanted to acknowledge both the funding sources and the people that really made this work happen. So we are funded by a NOAA OAR grant focused on omics, and we've also been able to leverage NSF support for these projects. And there's a research team that's really made all these results possible. All the graphs, all the sampling, processing, and the graphs that you'll see today are primarily done by Cynthia Becker, Laura Weber, and Brianna Garcia, and Krista Longnecker, Melissa Kiddo-Sewell, and Gretchen Schwar are responsible for the metabolomics powerhouse behind the analyses that you'll see here. So to get started, it's a little chilly out here in the Northeast Cape Cod today. I had to bundle up, but I thought we'd start off just going underwater and reminding ourselves what a tropical coral reef environment looks like. Coral reefs are indeed one of the most biodiverse ecosystems on our planet. About 25% of all ocean life passes through a reef at some point in their life, and it is the tropical stony corals that provide the framework that houses all of that diverse life. One of the paradoxes of reef ecosystems is that they reside in relatively low nutrient oligotrophic environments, yet maintain this incredible high rate of productivity. So that's something myself as a microbiologist that I've always been interested in, how microbes are contributing to helping maintain that high rate of productivity as well as biodiversity. Reefs are also economically important. Over 1 billion people worldwide depend on reefs for their livelihood or their economy. Economically, reefs have an annual net worth of $2.7 trillion each year, so they're incredibly important to coastal nations, island nations, including those of us that reside in the US, the Florida Keys, US Virgin Islands, Hawaii. I was just at the wonderful Flower Garden Banks reefs last week, and so I can definitely attest to the importance of reefs for our local economy as well. Unfortunately, coral reefs are under substantial change due to climate change, elevated incidences of disease, overfishing, and land-based disturbances. And so there's increased attention on these ecosystems, not only to understand the causes of the change by monitoring the ecosystems, but also to start to intervene and see if we can help reverse some of that loss and change. So now I'd like you to take just a minute, look at this photograph, and think about what really pops out at you. If you were down in this ecosystem, well, what is it that you would focus on? Maybe it's the corals, maybe it's the fish, maybe you're on the hunt for those snapping shrimp that are so loud. Well, when I'm down there, I'm thinking about the microbes. So they are indeed the most abundant life on this reef. They form association with all the macroorganism life that you see here, as well as they reside in the sediments and in the reef water. In just 1 milliliter, which is about 20 drops of seawater, there's about 1 million microbial cells there. So they're incredibly abundant. And I think one of the really neat, what I say, superpowers that microbes have is they are there because of the conditions, right? So microbes are there because of the specific pH, oxygen, temperature, organic matter, inorganic nutrients, biomolecules that are supporting their growth. So in that way, they're really sensitive indicators of the organismal as well as environmental conditions. Another superpower they have is they can grow really fast. So they divide on the order of once to 5 or even 20 times a day, depending on what their genomic capability is as well as the conditions if they're there to support them. My lab has been specifically focusing on reef water microbes. We study microbes in a lot of different environments, including corals, but we've been taking water samples for about the past 15 years. And we really like this approach because not only is it a non-invasive way to look at the reef, I mean, maybe I started it because permits were hard, but let's not go there. But we found that the reef microbial communities can be really indicative of what's going on a reef, as well as that oceanographic environment. So for example, reef water microbial communities are more diverse on high coral cover reefs compared to lower coral cover reefs, as well as highly protected reefs. And there's relationships between high coral cover reefs, as well as specific microorganisms that we find in the reef water. And other lab groups, around the world have found similar really interesting relationships on different reefs related to the reef water microbes as well. So I'm not going to go into that a whole lot today, but I just wanted to share that there's a whole backdrop to this microbial story in the water as well. So I think about the microbes underwater. This is probably what Liz Kujawinski, who's also on the call, thinks about are these dissolved metabolites. These are the compounds that are produced from metabolic reactions on the reef. They are the chemical compounds released by reef organisms as well as microbes. And primarily what we're going to talk about today is these dissolved metabolites that are indeed extracellular. So they are found outside the cell. So while each cell does indeed have metabolites in it, what we're talking about, what we're measuring are the dissolved metabolites that are not associated with the cells. These metabolites are important for energy and nutrient transformations on reefs, and they also serve as chemical cues or a means of communication between reef organisms for engaging in defense, reproduction, feeding, and a variety of other behaviors. What are these dissolved metabolites? Well, they can take on a lot of different forms. A lot of them we don't even know what the function are. Some examples are just vitamins, nucleosides, and indole-based signaling molecules. Studying these metabolites can really help us understand more about metabolic responses to specific conditions, as well as interactions between cells. And so the function the function or why these molecules are there is mostly unknown, the exact mechanisms producing them. But what's exciting is that with recent advances in mass spectrometry and extraction methods, we're starting to better understand the pool of metabolites in the ocean and on reefs. Now, some of these metabolites are taken up really quickly. They're very labile, and so they can have incredibly fast turnover rates, which can make them really sensitive indicators of what's happening in an ecosystem. So Liz Kujawinski, who's on the call with me today, has been really at the forefront of marine metabolomics methods, and her lab has developed novel analytical capabilities to identify and quantify marine metabolites. And microbes and metabolites are very tightly interconnected. So while macroorganisms associated with both of these two, microbial metabolisms, especially the microbes in the reef water, often depend on nutrient and carbon sources from the dissolved metabolites in the water. So these metabolites play a really important role in supporting the growth of microbes. And just examining these two parameters together, microbes and dissolved metabolites, can give us a really different look at reef functioning than has been typically achieved by divers going out on the reef and focusing on the macroorganisms. So we're looking at processes that are much smaller in scale compared to macroorganisms. And they can also be more sensitive to changes in that environment, given the fast rate of microbial growth and the very quick turnover of metabolites, and the fact that metabolites are indeed reflective of processes happening immediately on the reef. So given the fact that reefs are changing so much, we do need, and it would be helpful to have more sensitive diagnostic tools, especially non-invasive tools. We think that these two parameters together might be really helpful to further develop as a diagnostic tool for reefs. So what am I talking about? I'm talking about what I'd love is to develop a checklist of specific microbes and dissolved metabolites that we can look for in a reef environment, and that can be further indicative of the condition or the health of the processes happening on that reef. So that's the dream. That's where we'd love to go with this. It is complicated. There's hundreds of microbes and just those, hundreds of microbial species in just those 20 drops of water, and likely thousands of metabolites in that same volume. But I'm excited to tell you about today some of the progress that we're making towards this effort. So today I'm going to talk about four main things in my talk. First, I'm just going to give you a broad overview to some of the omics-based methodology that we use in our analyses. And then I'll tell you about some experiments we've done looking at the types of metabolites that benthic organisms are producing and how reef microbes are using those for their growth. And then we'll move to Florida's coral reef, and I'll tell you about a study that we've done examining microbial and metabolite patterns along that full spatial scale of Florida's coral reef. And then I'll just touch on some upcoming work that we have ongoing now, looking at temporal and disturbance-based microbial metabolite patterns on USVI coral reefs. So to get started, omics does enable investigation of coral reef microbes. You can see them under a microscope. You really can't tell what they're doing by looking at them in that manner. So how do we use omics? Basically, we go out on reefs and we first collect water, anywhere from 50 milliliters to 2 liters or even more is plenty of microbial biomass for our analyses. Here you see a diver with a Niskin bottle on a reef. That's one way that we collect water. We also send divers down with syringes if we want high resolution spatial sampling over different reef benthos. We've also put a groundwater pump down from a ship. So there's just a variety of different ways that this water can be collected. And then what we do with that water is we filter it to retain the microbes on the filter. So we get rid of the rest of the water. We just have the microbial biomass, and then we extract the DNA. So the methods in my lab are primarily genomics that we use to examine the microbes. So we extract the DNA, and then we do two different kinds of processes depending on the study. One is what I call the taxonomic microbiome. Many of you might be familiar with this technique where you amplify a marker gene. We use the small subunit ribosomal RNA gene or the 16S ribosomal RNA gene with primers that amplify both bacteria and archaea so that we can capture both of those microbial members. And then we sequence that, and then that gives us information about the taxonomy of the microbes in the water, as well as a look at the diversity of the microbes in that sample and compared to other samples. Another approach that we use in our lab is looking at the functional microbiome. This is also known as a metagenomics type approach. And so basically what we do is take those DNA extracts, we sequence them, and that allows us to look at microbial functional genes as well as assemble microbial genomes. So while this doesn't tell us exactly what the cell is doing, it tells us what genes are there and what the cells are capable of doing. And together, these two techniques can be quite powerful to get a nice look at the microbial community composition and its genetic potential. Now on the omics side of things, I'm going to next go through the processes that happen in Liz Kujawinski's lab. So she also goes out and collects water from the reef, generally 1 to 2 liters. Often we use a Niskin bottle for this just because it's free of some of the plastic additives that can leach and that do release metabolites and that can often mask the signal of a real reef metabolome. So what does she do with that water? Then filters it to remove microbes. So we're actually looking at different things. She wants to get rid of the microbes and just wants the water. So actually these are pretty collaborative techniques. We can both get something out of it. And then what happens with that water is that the metabolites are concentrated on these solid phase, on these cartridges here that you see in the picture. And then there's an extraction of those metabolites done using the solid phase extraction method. So once the metabolite extracts are in hand, there's 2 different techniques that Liz's lab uses. One is the targeted metabolome, and so this is where select metabolites are quantified using a mass spectrometry approach. I'm not going to get into the ins and outs of this method. Some of you probably know what all that means, and some of you don't. I'm happy to go over more of that later. But the thing to really remember here is that this method allows us to screen for a suite of known metabolites that have been quantified using standards on that mass spectrometer. And so this can be anywhere from 20 to 100 or more, depending on what's going on with a particular study. There's facilities that do targeted metabolomes, and it's a way to get comparable results about specific metabolites in environments and studies. The other method that Liz's lab does is untargeted metabolomics. So basically what this is doing is looking at all of the metabolites that are extracted in that sample, again using mass spectrometry based techniques. And what this is giving us are traces of each metabolite in the sample. So we don't exactly know what they are, but we get information about the mass to charge ratio as well as retention time. So standards could be run and or more methods used in depth to understand the identity of those metabolites. So this can be really helpful in trying to compare metabolite features between samples, and this can be run in both positive and negative ionization modes because some metabolites ionize in those different modes. So that's a little bit of overview to the methods, and again, happy to answer more questions or go into more depth later. But I wanted to get into some of the studies and tell you what we've been learning using these techniques. And the first study I'd like to share is the work we've done examining benthic organism metabolites patterns and how reef microbes are interacting with those metabolites. And so this is work that was started by Laura Weber. It's a former PhD student in my lab. She was a postdoc between Liz and I's lab, and she's now an integral member of the NSF CComp Center that Liz is director of as well. So there were 2 main questions that we asked with this study. How do metabolites differ between benthic organisms? Are they all making the same metabolites, or do they have distinct metabolites? And can we even measure them? There were a lot of unknowns going into this study. And then if we feed those metabolites to reef water microbes, do they grow on them? do we see differential growth based on the types of metabolites we give them? So how did we do this? This was based in the US Virgin Islands. We focused on 6 different reef organisms that are really common on the Caribbean reefs, 3 stony corals, Porites asteroides, Pseudodiploria strigosa, and Siderastria siderea. We also had 2 octocorals, Pluxara homoala, as well as Gorgonia ventolina. And then we also kind of added this in at the last minute because we were starting to realize how important, or unfortunately how important this is becoming on Caribbean reefs, especially in St. John, is this Ramacrusta. So this is an invasive crustose coralline algae that can overgrow and outcompete corals. It seems like larvae are not settling on it at all, and it's becoming a problem because it's becoming more and more of the cover on the reef instead of the nice pink crustose coralline algae. So what we did was take each species in a replicated manner and incubated that in microbe-free water for 8 hours with lights, and then we also had these control tubs that didn't have any organisms in them. And then after the incubation, we processed those samples to be able to analyze the metabolomes using both targeted and untargeted metabolomics methods. So first, I wanted to show you some of the data from the untargeted methods. And so here what you're looking at are non-metric multidimensional scaling plots. You're going to see several varieties of these in my talk today. And basically, if points are closer together in space, that means they have more similar patterns. In this case, these are metabolite traces from the untargeted method compared to points that are further apart. So here you're looking at all the different organisms, each individual plot, and the green plots, or the green circles, are the organism incubations, and then the orange are the controls. So you can see that the organisms are producing different metabolites than we're measuring in the controls, this was really good to see because we knew that we were able to detect metabolites that were being produced by organisms. I'm not going to show you this just because the graph is a little bit more complicated, but we also found a species-specific metabolite signature in each of these species. This work is now published in ISME Comms, and I think I have the citation there on the next graph. So I encourage you to look at that if you'd like more details. We also looked at the metabolites that were being produced by the different reef organisms using the targeted base method. And so this is where we ran the samples against a set of known standards, and we're able to quantify their abundance. And so what you're looking at here is a heat map. So if there is a red color, that means the metabolites, which are on the x-axis here, were more significantly enriched in the incubations compared to the control. And if it's blue, that means they were depleted in the organismal incubations. And so we have the different organisms listed here. I guess y-axis, I might have said axes wrong there. So metabolites are on the x-axis. So you can see that the variety of metabolites were produced by reef organisms. Those with the black stars are significantly enriched, and those with the white were significantly depleted. So there's quite a bit going on here, and I just wanted to point out a few more interesting elements of this rather than get into all the data. We did find, a variety of metabolites that were being produced by both the stony corals, the hard corals, as well as Ramacrusta. The pantothenic acid and riboflavin were 2 in particular that were produced by nearly all the reef organisms. These are B vitamins, which are important for enzymes and other processes. And interestingly, we've done some reef water metabolite studies over the reefs of Cuba and found that the reef water was indeed enriched in riboflavin compared to the surface waters. So riboflavin in particular might be a really good indication of life in an environment, which should not necessarily be surprising given the importance of B vitamins. Another interesting one that came out of this study, and this was a total surprise, was caffeine was being produced by some of these organisms and significantly produced by the Ramacrusta textilis, the invasive encrusting algae. So many of you might even be having caffeine right now, so most people are pretty familiar with this chemical. And so what's interesting here is Ramacrusta is so successful on these reefs, and we know that caffeine is indeed an herbivore defense, in many terrestrial plants utilize it in that manner. And so it's very possible that that is also something at play on the reefs, that caffeine is a deterrent to either grazing or other types of settlement processes. And then also indole-3 acetic acid was one that came up as pretty important in this study, and that was produced by Gorgonia ventulina. That is a plant growth promoting hormone, or at least that's what it's known. So it might be also playing important growth factors potentially associated with the algal symbionts in that CFAN. So that's just a little bit of a look at the targeted metabolome data, both the untargeted and targeted metabolome data. And then what we went on to do was feed the metabolites from Parides asteroides and Gorgonia ventulina to reef water microbes. So we did 2 different experiments, one where the coral metabolites were fed to reef metabolites, and then another one with controls where controlled metabolites from the control bins were fed to reef microbes. And then we examined microbial growth in both the light and the dark over 48 hours. This produced quite a bit of data, some fairly complex microbial interactions, and I'm just going to highlight a couple that I thought were really interesting to see. So we did indeed see microbial growth on some of the exudates. Some microbes didn't grow at all. Here's one that had really contrasting growth patterns on these 2 organisms. So what you're looking at here is the relative abundance of this particular microbe. This is a Rhodobacteriaceae known as HIMB11. So again, this was all from the DNA genomics-based methods, looking at the marker gene, small subunit ribosomal RNA, which is how we were able to quantify these relative abundances. So you can see over the 48 hours, we started with very little HIMB11 in this incubation, and then it grew to make up about 20% of the microbial community over the course of those 48 hours. And there were similar results in the light and the dark treatments. But when we gave it Gorgonia ventulina exudates, we saw hardly any growth. So maybe 1 or 2% of the community were these HIMB11. OM60 or NOR5 clade, this is a typical clade of marine bacteria. We saw a similar response to HIMB11. You give it Parides asteroides exudates and it grows like gangbusters. Again, 15 to 20% of the community there after 48 hours. And we saw a similar response in Gorgonia ventulina, where just a small percentage of those cells were able to grow in response to those exudates. So to me, this is like really exciting because, I always thought, microbes were ruling the roost in this environment, but actually they're, really responding to the metabolites that are available. It's definitely, controlling their growth. So here we saw microbial growth definitely differs based on coral metabolites, and this really has pretty wide implications for thinking about a lot of processes and interactions on reef, including related to pathogen recruitment and disease. So what did we learn from that experimental study? Basically, benthic species each release different types of metabolites. When you look at a reef and you look at a diverse reef, think about what each organism is doing and how it's contributing differently to that ecosystem, which I think really holds to the point that having ecosystem diversity means you have chemical diversity and microbial diversity as well. We found that corals produce B vitamins and a variety of other metabolites. So we have some good starting points of what a coral-rich reef might look like from a metabolite standpoint. And we also showed that benthic metabolites directly impact microbial growth. So now I'd like to move on to the next research study that I wanted to share with you, which is more of an observational study. And this was really spearheaded by Cynthia Becker, who is a PhD student in my lab. She's going to defend in about 3 weeks and has just done a really fantastic job on this study, and as well as another one I'm going to tell you about in a minute. Basically what she's been doing is looking at microbial metabolite patterns across Florida's coral reef. This is a collaboration with Mote Marine Lab Aaron Muller and Abigail Clark, as well as Stuart Sandin's lab at Scripps Institution of Oceanography. And basically, OceanX came to us with an opportunity to spend 3 weeks on Florida's coral reef and work in coral health capacity. And so we found this to be an amazing opportunity to apply our omics-based tools to learn more about this ecosystem. So we had access to this Alusha vessel, and so over the course of those 3 weeks, we visited 85 reef sites throughout the Florida Reef Tract, which you're seeing here are divided into the different 8 zones. These are historically placed zones, which also relate to some of the habitat and environmental characteristics. So we went from Zone 1, North Key Largo, Biscayne Bay, throughout the upper, middle, lower keys to the Marquesas, and then these even remote Dry Tortugas National Park reefs. So this cruise was done in June of 2019. Throughout the reef, we had throughout the reef, before I get into the omics, I wanted to share some of the data from what the reefs looked like. So for 45 of the reefs, Stuart Sandin's group did photo mosaics, and we have these just like amazing now maps of all these different reef sites. When we look at coral cover on those reefs, we find that it does vary by zone. So these graphs that you're going to see here are color coded according to the map here, and you can see that reef cover, coral cover, hard coral cover is variable throughout the reefs, and dry tortugas is consistently fairly high, around 15 to 20% compared to, for example, Biscayne Bay, North Key Largo, which is hovering less than 5%. But there were a few reefs with relatively high coral cover mixed into that reef tract. With Aaron Muller's team, we were able to get a really good idea of what was going on with the stony coral tissue loss disease. So most of you might, or some of you might be familiar with this. Some of this might be new to you, but this is a lethal, contagious disease that has been spreading throughout the Caribbean. It started outside the port of Miami. In June of 2019, when we sampled, it was down in the Marquesas. Here's disease prevalence, and it was not yet in the dry tortugas. So one thing nice about this data set that I'm showing you is that we do have one site that was not affected by this disease at that time. And it was really quite high in zone 5 and sort of the middle lower keys at the time. Here's just a couple of photographs of what this disease looks like. It has had a significant impact on coral mortality in the Florida reefs, as well as other reefs in the Caribbean. Before I get into the omics data too, I wanted to share some of the other parameters of the reef that we typically measure from reef water, heterotrophic bacteria, as well as total organic carbon. So this was done for all the 85 reef sites that we visited. And so you can see some really nice patterns that are related to the zones. Heterotrophic bacteria does increase across, or decrease, I guess, if you're going from Key Largo to Dry Tortugas. So that significantly differs with reef zone, and that's related to total organic carbon concentration too, which tends to be lower in the Dry Tortugas, higher in the middle upper keys, as well as Key Largo, Biscayne Bay. So we did see some relationships between these environmental parameters as well that related to the reef zones. Now getting into the omics data, I'm going to show you a lot of these graphs of just dots, dots and plots, and these are principal coordinate ordinations when they're just the plot, just the dots. And again, what you're looking at here is when the dots are closer together, the microbiomes are more similar. So this is the taxonomic microbiome. So this is when we use the 16S based analysis to get at the identity and diversity of that community. And so you can see that lots of different colors are clustering together, showing you the strong zonal zonation of the microbiomes at these different reef sites. We measured a lot of other parameters of the reef, both reef cover as well as environmental features. And when we examine those in relation to the plot using a distance-based redundancy analysis, we can see the relationship of the different reef parameters. And this is a per-MANOVA analysis showing you further the relationship between the reef parameters and the taxonomic microbiome. So I'm only showing you the parameters that were significant here, but you can see reef zone has the highest F value, meaning the strongest relationship to the taxonomic microbiome. So that is definitely a strong indication of the reef, as well as total organic carbon, Prochlorococcus, which are photosynthetic bacteria, heterotrophic microbes. These are abundances that we measure using a non-genomics-based method. Synechococcus abundance, the prevalence of stony coral tissue loss disease, as well as the abundance of pico-eukaryotes, hard coral, and soft coral. So what's nice about this is that from these reef water microbes overlying the reef, we're getting an indication of factors that are related to that microbiome features that are related to not only that environment, which is related to the oceanographic environment, the biogeographic environment, as well as some of the reef features itself. So it does seem to be fairly comprehensive in manner. I will mention we only did this analysis for 27 of those reefs. We would have loved to do it for 85, just there wasn't enough hours in the day, enough people on the ship. But we were able to get quite a few samples. We were able to get samples throughout the distribution of this of Florida's coral reef, and these were done in duplicate at each of the reef sites. Samples were taken in duplicate. We also looked at the functional microbiome. So this was done using the metagenomics-based approach where we're looking at microbial functional genes. And this produces a ton of data. So basically 2 orders of magnitude more data than I showed you with the taxonomic microbiome. So there were 14 million reads per sample. This was also done in duplicate for samples taken from 27 reef sites. So what's interesting about it is we see basically a very, very similar ordination pattern to what we did here with the taxonomic microbiome. We see samples that are clustering in basically the same area, primarily by reef zone. And the way that we looked at this, the functional genes too, is we took out the bacterial functional genes and compared the relative abundance of those. So there were 592,000 bacterial functional genes that went into this analysis. Whereas this taxonomic microbiome, we're just looking at one gene. So it's really interesting to us that we see the same pattern with like 2 orders of magnitude less data and I don't know, hundreds, not hundreds, tens of thousands of less dollars too. We can get a pretty similar glimpse of the microbiomes. When we overlay the other features that we measured on the reef, there were also a number significant less than with the taxonomic microbiome, but some of the same major players. Reef zone was again the strongest parameter. Total organic carbon, Prochlorococcus, heterotrophic microbes, Synechococcus, pico-eukaryotes, and then disease prevalence. Next, I'd like to show you some of our metabolome results. So this was done for even fewer reefs just because of the time-intensive nature of the sampling, and these are also not cheap to process. So 13 reefs, but again, we were able to take samples throughout the distribution. Each reef, we took triplicates this time just because we know a lot less about variability of metabolites on reefs than we do even microbes. And so you're looking at the metabolites from 39 targeted metabolomes, so 39 metabolites that we quantified in this study. So you're looking at each dot is representing the relative abundance of those 39 metabolites. So also some zonal based colored clustering in this graph. When we overlay the features, there's actually quite a few that are significantly related to the taxonomic metabolome. Just like with the microbiome, reef zone was the most significant, most related, but then we have a variety of both reef, microbial environmental, as well as reef-based features, so the soft coral and the sponge and the hard coral. And so actually about as many features were significantly related to the targeted metabolome as the taxonomic microbiome. So that's interesting because both of those techniques of the 2 genomics and metabolomes methods are the they're cheapest. they cost the least. So that's really interesting to find from I guess, applied standpoint as well. When we looked at the untargeted metabolome, we looked at both ionization modes. I'm only going to show you the negative mode here. So there are about 1,400 features that are displayed here per sample point. Again, we looked at this on the 13 reefs in triplicate. Again, we saw zonation-based patterns in this untargeted metabolome, and there were fewer numbers of significant related features. Reef zone was, yet again, the major significant feature related to these untargeted metabolomes, as well as some of the more microbial related parameters that we measured. So this was really exciting just across that data set to see how 4 different omics approaches all differentiated reef zone. And, we expect that both microbes and metabolites are related to oceanographic, land-based environmental, as well as reef-based features. And it does seem like we're capturing all of that in these kinds of data. So there's quite a bit here with the study. I'm not going to get into much more other than I wanted to show you this summary diagram that Cynthia put together for the paper. And I haven't explained all of these things even that go into this, which hopefully you'll be able to read her paper soon and see. But what I wanted to point out is that with the metabolomics and the genomics, you're able to get a really different look at these reefs. So yes, going out with the photomosaics and the disease surveys, you're getting an indication of what the reef looks like for 2 different spots, such as dry tortugas with a more coral cover and diverse reef compared to North Key Largo, Biscayne Bay, that has more of a diseased lower coral cover reef. But when we look at the water and you apply genomics and metabolomics, it looks really different. And there's some specific components that we can point to. Some of them we know mechanisms that are causing differences, and some of them we still need more research and experiments to understand. But to me, it's really exciting that we see these unique microbial metabolite signatures in the reef water, taking a non-invasive sample that really takes less than a minute to obtain. There's quite a bit we can learn. So what did we learn from studying Florida's coral reef with our omics techniques? We found that all these techniques differentiated the reefs according to reef zone, which was really exciting. These are historical zones based on not only biogeography, but there's some documented reef differences there. We also found that microbial related features were apparent in all of those environments and significantly related to the genomics and metabolomics approaches. We found that overall the taxonomic microbiome and targeted metabolomes were most related to environmental and reef features. So like I said, these are also the cheapest of the 4 methods that I'm presenting here. So that's exciting for a variety of standpoints, and also because we know more about, there's more data on taxonomic microbiomes. We can put an identification on a targeted metabolome or metabolite that we're analyzing. So there's more opportunity to go in there and understand what exactly is happening and why they're more or less related to those features. And then overall, we think, this data set is really showcasing how microbes and metabolites provide a really unique signature of reef health that you just can't get by putting a diver in the water. So lastly, to finish up, I just wanted to touch a little bit on some of the ongoing studies we have in St. John, U.S. Virgin Islands. So we've been studying 8 reef sites there since 2016, applying some of these omics-based methods. So this is primarily focused on the south shore of St. John, where it's in the national park, so free from direct land-based influences. One of the studies that Cynthia Becker has been working on is going on these reefs every time we're there, taking water samples and looking at the microbes using the taxonomic microbiome-based approach. So the timeline you're seeing here in blue are all the opportunities that we sampled, and then we were able to capture samples before and after the hurricanes Irma and Maria that came through, as well as once stony coral tissue loss disease unfortunately emerged in that ecosystem. So this is a fairly unique sort of natural-based experiment that we're excited to hopefully share soon some of the things that we're learning in St. John. And then another exciting piece that I wanted to share with you is Brianna Garcia is a very talented postdoc in Liz Kujawinski's lab who's helping us improve methods to even detect these reef metabolites. And so, you know, behind all these omics techniques is sort of the little notion that you're only measuring what you can, what your methods are detecting, right? And so for metabolites, this is kind of a scary thing to think about because a lot of methods are not able to capture some of the metabolites that we might be most interested in. So in particular, low molecular weight polar molecules that may be really important for reef health and functioning. We think we're missing a lot of that. So what Brianna has been doing is applying a derivatization-based method to improve the detection of these low molecular weight compounds, and basically what she's doing is just adding an additional molecule to these smaller molecules, which allows them to be captured on the solid phase column for extraction. So she's been applying this to a short-term temporal study in U.S. Virgin Islands. And I'm not going to get into much of this other than to say that, hey, it's working. We're really excited. Brianna, you're doing awesome. So here we're looking at different metabolites that the targeted method is quantifying. And we're comparing those metabolites from a variety of different reef water studies, including the Garcia method and PrEP. And so there's at least 24 novel metabolites that Brianna is able to detect with this method. And we're really excited to see how those vary with different reef health parameters. So a little bit of upcoming look at what's happening from St. John. So lastly, I'd like to just summarize what we have presented here. So I want you to take away two main points today. One, when you think about when you're underwater, thinking about reef health, think about microbes, think about metabolites. This is a really important relationship between the benthic organisms, what's there, the metabolites that they're producing, and pelagic organisms too. Fish are producing metabolites. It's not just benthos, and the microbes that are existing there. This has important implications for the health, the biodiversity of the ecosystem, the resilience of the ecosystem, and the susceptibility of that ecosystem to disease. So these are hidden components of the ecosystem that Omics is helping us unravel and start to look at. And the other thing I want you to think about is diagnostic. So I didn't talk about this too much, but this is where we're going with all of these studies. So we think microbes and metabolites provide this really unique view of coral reefs and reef processes, and might provide a more sensitive indication of reef change, maybe at a time when there's still when there's still time to go in and do something about the problem. So that's where we'd love to move some of this work. How are we going to do that? Here's just a couple of the next steps. Is identifying key diagnostic microorganisms as well as metabolites that really signal different aspects of reef health. That's something these studies are all helping us get there. We still need more observational and experimental studies to further build that. And second, we're really interested in moving towards more automated sampling and analysis platforms. And Liz Kujawinski is already kind of thinking and working in this space, but these are just a couple of examples of where we could go with this. You know, I know AOML with NOAA has already made an eDNA sampler. The genomics methods I'm describing could easily benefit from these kinds of samplers, which could be placed out on reefs. It seems like very cheaply. And then the other idea would be robotics, like the Curie robot, which is able to take additional payload onto that, including a sampler, and go out to a unique environment and take microbial and metabolite samples for us. So I think working towards getting more samples will only help us with number one of better understanding the key diagnostic indicators. And lastly, I'd just like to acknowledge the key players in this work. We have funding from NOAA, OAR, NSF, and OceanX. Elizabeth Kujawinski is on the call for questions. She is responsible for all of the metabolomics that you saw today, or directing all of it. We have a wonderful primary research team that has made all the wonderful graphs and analyses that you've seen today. And then support staff at Woods Hole, Moat Marine Lab and Scripps Institution of Oceanography also contributed to this as well. I'd like to acknowledge our permitting agencies. And lastly, hopefully you have some questions for us. So please put those into the chat. Thank you.
Awesome. Thank you so much, Amy. That was a fantastic presentation. Yes, we do have questions that have already come into the chat. So folks, feel free to type those into the questions box and we'll read them out loud for Amy and Liz to answer. So our first one comes from Sherry. So Sherry actually has two questions, so we'll just do one at a time. They say very interesting responses you saw with the rhodobacteria and the Gorgonia ventolina metabolites. The first question is, do you see any pattern in the types or species of corals that grow in close association with each other based on potential metabolic activities that may limit potential pathogen, bacteria or algal impacts?
Yeah, Sherry, that's a really great question. And I don't have an answer for you. I think that would be an amazing study to be able to kind of, bring together some like high resolution, either survey or photomosaics and metabolites, as well as microbes, really to understand, what the spatial extent of those interactions are. And I think there would have to be a temporal element to it too, because I think it's going to be changing depending on what those corals are producing, what time of day it might be. So yeah, that's definitely a next step. And we have characterized microbes like close, over different species of corals. This is some work that Laura Weber did, and we do find microbial communities, differ in that reef water over the corals, and that's kind of where our Exudate experiment was going, trying to quantify that in the lab. It's a little harder in the field, but yes, it would be wonderful to look at a community assemblages and see how we can build more resilient microbes. Great question.
Thanks, Amy. And this is the second question from Sherry. They ask, do you have any plans to attempt to identify or isolate the metabolites from Gorgonia ventolina in an attempt to provide therapeutic relief to corals that may be more susceptible to bacterial or algal pathogens?
Yeah, that's also a great question. It's not something that we've thought about, but I'll hand it over to Liz too if you want to talk a little bit about. I mean, there's definitely relevance for some of the things on a therapeutic standpoint that we're talking about today.
Yeah, this is Liz. Thanks a lot for the question. What I would say is that moving from the untargeted metabolome data that Amy summarized in the slides to an identification based on isolation and then into therapeutics is a colossal challenge. And just the identification piece alone is a pretty significant bottleneck. What we're finding at the moment is that the databases that are available for comparative analysis leading to identification are highly biased towards the human system. And consequently, we have a hard time finding our marine samples there. So I think there is definitely room for improvement and room for growth in that area. But at the moment, it is a bottleneck that we're working with other types of analyses to try and circumvent. Thanks.
All right, thank you both. We have a question here from Elise. They ask, based on your summary plot, would you say that the Synococcus to Prolochoracoccus ratio may be an indicator of reef health? Excuse me if I pronounced anything wrong there.
Yeah, that's a great question too. So those two cyanobacteria, like we know from many oceanographic field studies that they tend to have different distributions based on whether you're in more coastal or oligotrophic waters. And we've seen some pretty interesting trends in some of our reef water studies too, where Prochlorococcus, for example, is highly abundant on more forereef, healthier type of environment. So I do think that they play a role in water quality type indicators of water quality. But I also think that we don't know about their true potential as indicators as well. What else are they indicating? So that, you know, we've seen some Prochlorococcus, for example, grow on exudates from corals, and that's not something we predicted was going to happen at all, given their oligotrophic nature. So I think there's a lot of potential there, and I do think that I'm starting to think of them as different health indicators of reef. I just don't know exactly what all they're indicating, other than some basic nutrient parameters. So thanks, great question.
Thanks, Amy. And we actually have a question from Nicole. So Nicole, if you want to come off mute and ask.
Hey, Amy. Hey, Liz. Great talk. So my question is, what spatial scale, ideally, should these micrometabolite reef signatures be applied to in an ideal setting?
Yeah, that's a good question. So we've like I showed you in the Florida data set, how, you know, we took some duplicates and even triplicates for metabolites on reefs, and when we take those, we take them in different places on the reefs. And I think this is kind of what you're getting at is, you know, do you need 100 samplers? Do you need 1,000? You know, do you need just one? So it does seem like, especially for the metabolomics, there did seem to be more variability depending on where you were on a reef. I don't have an exact like spatial scale recommendation right now. I mean, we're still at the point where yes, more data per reef would be better, but you know, at least when we do the genomics-based method, like I can tell reefs apart based on their genomic signature, even if samples were taken on different parts of a reef. So you know, if you had limited funds and just kind of wanted to get going on something like this, I think one to two samples per reef would be ideal to start with. And, you know, what is that spatial extent? Well, I mean, it's definitely not going to be kilometers. Yeah, most likely tens to maybe hundreds of, or probably more like tens of meters is what it's integrating.
Thanks.
I don't know if I satisfied that or not, but.
All right. I don't see any other questions coming in from our audience members, but if you have a lingering question later on, you know, please reach out. You can reach directly out to us actually at noaa.omics@noaa.gov, and we can forward your questions on to both Amy and Liz. But yeah, so I think with that, we're going to wrap this up. So Amy, Liz, thank you both so much for being here today, for presenting and for answering questions. We really appreciate it a lot.
Thank you all for being here.
Thanks for having us.
And thanks so much also to our audience for joining us today. The NOAA Omics Seminar Series normally takes place on the third Wednesday of every month, although we do schedule seminars outside this time. So just stay up to date on what seminars are coming up by checking out our Omics website. Link to that in the chat now. So thanks everyone, and we'll see you at the next one.